Hello and welcome to the next half paper. It should have been about 45 minutes. Um, Again, this is my top tips video. I shall send the mark scheme as well. First of all, let's just have a look at the top tip on this question is just read the information. It tells me in the question that there's a mutated virus. It's a mutant strain, and therefore that's what I've got into my answer. Why is it higher afterwards? Well, B and D are higher because the memory cells for those have been activated. And why is there no A and C? Well, because the virus has mutated, so it no longer has those antigens. They've changed enough that they no longer fit into the receptors of those memory cells, so those memory cells are not activated. Nice and easy. 1.2 is just cytokines. Like what's two T helper cells for 2 marks. It's also worth knowing that it's the T helper cells that get killed by HIV as well. That's also on the syllabus. And then it shows us this antibody. I'm just making sure here that I'm comparing both in my answer. Yeah, it's got 3 binding sites, not 2. So that's my second answer. That there's 3 antigen binding sites. The normal antibody has got 2. And if you see, there's a inward triangle, and then a more rounded shape here. So it's got different binding sites. I could also go that there's no constant region where you could attach other things to, like monoclonal antibodies do. And then how do you get a false positive? It's all about washing. You add your stuff to the well, and you add the last antibody you add carries an enzyme, and that antibody is supposed to bind. And if it doesn't, and then when we wash it, we remove unbound antibodies. Well, if there was nothing for it to bind to, it should be removed when we wash. However, if it was ineffective washing, it will be still present. I just have to get into a positive test. So the enzyme will be present, therefore it would react with the colourless substrate and you'd get a colour change, and that's the positive result. So that's 1.4. Then we just get to a nice straightforward speciation question. And crucially, these tigers are in different parts of they are in different geographical areas. Those in Sumatra are cut off completely from the mainland, and therefore, what kind of speciation is it? Well, there's 2 types. There's sympatric, I think same, and allopatric is the one where it's geographically isolated. And I say both. It's allopatric speciation because they are geographically isolated. Mentioned that there's no gene flow between the populations, so they're kind of cut off from each other. And then in the different areas, you get different selection pressures, could be different climate, etc. Whacking genetic drift as well, because that just gives you a randomness as well. So they're going to evolve in a different way, so you get different allele frequency, and then eventually they become so genetically different that they can't breed to produce fertile offspring. It's a nice, easy question. I think is that if you know your biology. Then we get to a Hardy-Weinberg. They always try and give you a hint to get to Q squared. 1 in 2,500 babies are born with cystic fibrosis, and it is it's telling me what's recessive because it's told me that a couple who are both carriers for the condition have a 25% chance of having a cystic fibrosis. So it must be recessive allele disease that you might know anyway from GCSE. So 1 over 2,500 is Q squared. That's a frequency of homozygous recessive. Square root it to get Q. P is 1 minus Q, so that's 0.98. That's the first marking point. Just make sure that you put words in your answer. That a carrier is 2PQ. If you just put 2PQ equals, then the examiner doesn't know that you are working out what the carrier is. So make sure you do that. So then 2 times P times Q is 0.0392. It asks me for the number of people to 2 sig fig. So I have to multiply my frequency by the population size, which is in the question up there. So it's 2.6 times 10 to the 6, or 2.6 million, or 2,600,000. Then we get to colour blindness. see, and this is I put on like the working out that I would do in this question. It's asking about 13, and therefore I need to know its parents, and its parents, I didn't have to do number 3, but I was showing off, and it's an explain question. A lot of people in your mock exam described this kind of question. I have to say, how do I know it's come from number 2? So yeah, start at number 13. Number 13 is male, so must inherit the Y chromosome from father number 8, and therefore must get the X little a allele from its mother, his mother. Again, a lot of people look at number 13 and think, oh, you've inherited your dad's colour blindness, but that's impossible because males inherit Y chromosomes. And then number 7, must inherit the X little a from number 2. I could either say, because number 2 is homozygous recessive, or I can say, it kind of come from number 1 because he's got normal colour vision and therefore must be X big A, and therefore it must come from number 2. Again, there's lots of ways of saying it, but I have to say more than it must come from number 2. It is a because in the answer, because it's an explain question. That's that one. So I should zoom this around. So let's zoom over here. And then we get into dihybrid. I've done my working out as I read it through. A dwarf pink flower plant was crossed with a heterozygous tall white flower. They love doing this because if you're heterozygous, you must have one of each allele, and therefore you must have a dominant allele for each gene. And therefore I know that tall is dominant and white is dominant because a heterozygous plant would have a dominant allele for each gene. And then I do my working out. So I've done all the hard work as I read it through. I've done my working out in the question. I can then work out the genotype of the offspring, put it in order, phenotypes in the same order as the genotypes, and it's a 1:1:1:1 ratio. And then we get into another species of plants. All right, so it's another species. That means its genes will work in different ways. It's got 2 genes controlling just flower colour. Right, so that's epistasis. 3.4 is epistasis. And then what are the possibilities for being pink? Well, to be pink, you'd need a normal enzyme. So I need a big A dash, and then it must be homozygous recessive for the B gene because it makes non-functional B enzymes. So it's little b. So all the possible genotypes are big A, little b, or heterozygous on the A and little b. Again, get those questions right. They are relatively tricky. What have I done here? And I'm back again. And then we've got a white flower with the genotype little a, little b is crossed with a flowering plant that's double heterozygous. The table below shows the F1. I've just done a bit of working out at the side, and I encourage you to do the same. So I'm doing a cross between, this individual there that will make little a, little b gametes, and this one here that will make all of those gametes. So I'd expect a 1:1:1:1 ratio if it showed complete segregation. However, I'm getting nowhere near that. So these match these. But these numbers are vastly different to what I'd expect. So this just means it's linkage. So these 2 genes are on the same chromosome, so it's gene linkage. The red parent plant, again, that's the only one I can talk about because it's got different alleles of genes, has, and see, the 87 and 90 must have come from these genes over here that I'm circling again. Sorry, those gametes over there. So that tells me that on the parental chromosome, so there's on my pink parent, it's got a homologous pair of chromosomes, and the big A and the little b are on one, and the little a and the big B are on the other. So it means it's like that. So it's going to make lots of those gametes after meiosis because it doesn't need crossing over to separate those alleles of genes. However, we are getting the odd recombinant, top and bottom one. Therefore, crossing over is occurring rarely to produce my big A, big B, and my little a, little b gametes. So that's that one and that one. on my little working out table. And because there's so few of them, I know that these two genes, these here, are very close together because I'm very rarely crossing over at a locus between them. So they are generally inherited together. Gene linkage is very difficult, I think. Again, they put it on homework. People just don't do it because they don't know it. But there is if you do some working out, you can work out what it must be. And it's just knowing if it is gene linkage, then it's quite easy to spot because you get those weird numbers that are produced. And then I've got a third species of plant, so I have to do a chi-squared on this. So I've got 4 different genotypes. So 1, 2, 3, 4. Therefore, I've got 3 degrees of freedom, and we work at p equals 0.05, so my critical value is 7.82. So therefore, I write chi-squared 7.92. My decimal place could look a bit better. Is greater than the critical value. That's critical. That I write greater than. Therefore, we reject the null hypothesis. If chi-squared was 0, there'd be no difference between observed and expected. Here, and as the difference between observed and expected gets bigger, then I become less confident that it's following the same as expected. And therefore, if that sentence says greater than, this says less than, so they're always opposites. 5% comes from my p equals 0.05. Probability comes first, that the difference between the F1 numbers and those expected is due to chance. So for this plant, if these genes were independent of each other, then I'd expect a 1-to-1-to-1 ratio. However, I've got lots of those here. I've got 28 of them. So this looks like this is a plant that the heterozygote would have gene A and gene B on quite a distance apart. So the chance of getting the recombinants, that one and that one, is actually quite high. But he's not asking me about that anyway. I can just say what I've written down there. And then we get into a how science works question. So the assumption we have is that water in equals water out. Or I could put, I can't put that 0 water will be used in photosynthesis because I'm also making the assumption that the plant is well watered and water isn't going into the cells and making them turgid. So it needs to be what we've put there. It'd be too vague anyway. Find the anomaly. We just zip across these rows and look for anomalies. Yeah, and there it is on 57 in trial 4. Tick. And then to what extent is it? Well, just work out the mean without the anomaly. So then it's 29.4. They've got 34. And if I do the difference divided by the original times 100, I get a 57.6% increase in the mean. So that mean is 15.6% too high because I've included an anomaly. We have skewed the mean. And then we get to this maths question that people do struggle with because there's information elsewhere. And I should have pointed out where I was here. Yeah, like numbers in questions are important. And there you can see, like there's my 5 minutes. This distance moved in millimeters in 5 minutes. So and that's the reading I'm taking from there. So I'm writing down from my table that it moves 86.2 millimeters in 5 minutes. Therefore, I can get one easy mark just by dividing by 5 and saying that it moves 17.24 millimeters in 1 minute. So that should be easy. And then use pi r squared. It's told me that the internal diameter of the capillary tube is 0.7 millimeters. So the radius is 0.35. So I do pi r squared. Pi, there's my radius of the internal diameter square multiplied by my answer over here. And that's 6.63 or 6.6. It doesn't say what to round it to, so I can do it to what I want. And then why do we have a control? Yeah, so it's to compare the other experiments to show that any change investigated is due to the change and not to anything else. There they've used a calibrated pipette. Why would a capillary tube be better? Well, they have a smaller diameter, so you get higher resolution, so less uncertainty. Yeah, my scale's going to be taller. If it was a capillary tube, I get a bigger change for the same amount of volume. And we're almost there. Oh, flown through. And then energy and ecosystems. We have to use the diagram to calculate percentages of sunlight energy that will be transferred to feces. Well, that's here. So it's the percentage. So I'm going to start with 100 and then apply the 2.1, the 12.7, and the 58.2. So it comes in at 0.155% to three significant figures. I shouldn't need a percent on the end. And then in this ecosystem, the net productivity was 24,525. Use information from diagram to calculate the gross productivity of the zebra. It's only worth 1 mark. It should really be 2, I think, this, because it seems a lot to do for 1 mark, because I have to know what GP is, but it might just be a hard question. So first of all, I'm going to just find out how much energy gets into the zebra. What goes into the zebra's mouth? It's 3,114.7 kilojoules per meter squared per year. So I've just used my 12.7% of the vegetation. GP is the energy in the nutrients absorbed. So it includes, basically, I have to knock off 58.2%. So the rest of it is going to be absorbed. So it's 41.8% of 3,114.7, which is 1,302. That's my answer. So GP isn't what goes in your mouth; it's what goes in. It's what's absorbed via. If this is an animal, and it is a zebra, it's what's absorbed via its ileum, as in the energy and the nutrients absorbed. And then why does only 2.1% of the energy striking the leaf is available? Again, loads of stuff you can write here, but these are my favourite 3. Light that is the wrong wavelength, so it's reflected. I can't just put light is reflected. I have to say about wavelength. Sunlight energy misses chlorophyll, so it's absorbed by water, and that does hit chlorophyll. Photosynthesis is inefficient, and there's heat loss. And then I get onto my 5 marker, and I put the fixed law at the top just because it's how I frame my answer. So it's fish gills, and it's the concentration gradient multiplied by surface area divided by thickness of barrier. So I start off first of all by getting my surface area. So gills have a large surface area, as each gill has many filaments that each has many lamellae. There we go. So I've got 2 surface area things. Each lamella is covered in a dense capillary network. That's another surface area thing, which contains blood to absorb oxygen and flow to maintain the concentration gradient. So I've got my concentration gradient. Water and blood flow in opposite directions, so there's a counter-current flow mechanism to maintain the diffusion gradient along the entire length of the capillary. That's my stock answer. And then I just need my thickness bit. So the epithelial cells that form the capillary wall, that's trying to be an R, are thin cells, and the capillary wall is only 1 cell thick, so a very short diffusion distance. Easy 5 marks. Happy days. Hopefully more people will see this video than the last, but it's only taken me a few hours to write all my answers in, and 18 minutes to record this. Joy.